focus of this video is on stem cells. We want to understand what stem cells are, what are the different types of stem cells, where do we find them, and why are we so interested in them? You know, what's their therapeutic significance to us? So first of all, what are stem cells? Well, stem cells are undifferentiated cells. They're not specialized cells. They divide by mitosis, and the daughter cells that they produce differentiate into specific specialized cell types, right? But they themselves are in an undifferentiated state. And we already talked about one important type of stem cell that we see, and that's the embryonic stem cell. And that leads us into one important function that stem cells carry out, and that's driving embryonic growth, right? So we already talked about this a little bit. We talked about the fact that these cells in the inner cell mass at that stage of development that we call the blastocyst stage following cleavage, those cells are embryonic stem cells. And they will go on to form the embryo. Each one of the cells present within this region of the blastocyst can become any type of embryonic tissue, right? It hasn't differentiated at all. All possibilities are still open to every one of those cells, and therefore they are referred to as being a type of stem cell that's referred to as being pluripotent, meaning that it can become any type of cell in the embryo. But now remember that we're talking about a very early stage of embryonic development. We're talking about roughly 5 days after fertilization that we see these embryonic stem cells in this undifferentiated, completely undifferentiated state. Because we know that what happens following this stage of embryonic development is we get that progression of events called gastrulation. And in the process of gastrulation, we see formation of those 3 germ layers, right? So we see differentiation occur into one of 3 primitive tissue types, either ectoderm, mesoderm, or endoderm. So within about 2 weeks following fertilization, those cells have already differentiated and become specified to a particular tissue type. But again, what's causing the cells to be undifferentiated at this stage of development, but differentiated at this stage of development, are the sets of transcription factors, and therefore the sets of genes that are being expressed at these different stages. So here at this blastocyst stage, all of the genes that need to be expressed in order to assume an identity as ectoderm, mesoderm, or endoderm, those are all repressed at this stage of development, right? Once they become activated, we see differentiation into those different tissue types. But at this stage, all of those genes are repressed, and they're repressed by specific repressive transcription factors that are expressed here, but not here. But at this stage of development, they're still pluripotent. They can become any embryonic tissue type, right? They're not limited at all. Right? That's why they're referred to as being pluripotent. So they're obviously very important during the process of embryonic growth and development, because they are the source for those billions and billions of cells that get produced during embryonic development. In addition to those embryonic stem cells, we also have adult stem cells that would be found past embryonic development and that actually persist into adulthood. And these stem cells are really important for tissue replacement. So we have some types of tissues that are constantly sort of dying off and needing to be replaced, including our blood cells, our tissues like skin, the lining of the stomach, for example, constantly needing replacement cells generated. So we would have stem cells within those specific tissues constantly generating a set of replacement cells to replace worn out tissues. We also need them in the case of injury for tissue repair, right, to generate new populations of cells. In contrast to the embryonic stem cells that we just discussed, though, adult stem cells are not pluripotent. That is, they can't assume any cell fate, or the products of their cell division can't assume any cell fate. Instead, they are either what's referred to as being multipotent or unipotent. Right? Unipotent means that they can only produce one cell type. Right? Examples of that would be those stem cells that replace the skin and the lining of the stomach. Those stem cells can only produce that particular type of differentiated cell. Multipotent can produce a few cell types, but the key thing is that those cell types have to fall within the same tissue type. So examples of multipotent stem cells would be something like the bone marrow stem cells. So these are stem cells that exist within the long bones and the flat bones in the adult, and they produce red blood cells as well as all of the different types of white blood cells that circulate in our body. So red blood cells that carry oxygen, white blood cells that function in immunity, they all come from stem cells within the bone marrow. But bone marrow couldn't, for example, make skin cells, right? So the options are limited. It's only within that tissue type that bone marrow stem cells can generate different types of cells. Neural stem cells would be another example of a multipotent stem cell type. So once upon a time it used to be thought that, following development after early childhood, you had the full set of neurons that you were ever going to have in your life, and that over the course of your life all you did was lose neurons and you could never regenerate any. We know that that's not true now. We know that there are stem cells that exist within the brain, the central nervous system, and those stem cells can be induced to divide and produce new neurons, different types of neurons, of several different types, right? So we do have a limited capacity to regenerate, to some extent, brain tissue, right? These neural stem cells would only be able to produce different types of neurons, right? You wouldn't find neural stem cells making white blood cells, for example. So their options are limited. So they're referred to as being multipotent but not pluripotent. And again, unipotent adult stem cells can only make one cell type. Cardiac muscle tissue, we also have stem cells within that tissue, and that would be another example of unipotent stem cell. Cardiac muscle stem cells can only produce differentiated cardiac muscle cells and no other cell types. So it seems like if we know that these stem cells exist within these different organ systems, then if we had some clinical condition that resulted in the death of tissue, then we should be able to just stimulate those stem cells to reproduce and repopulate and repair or replace that diseased tissue. So for example, if you have a disease like Parkinson's disease. Parkinson's disease causes the death of a specific set of neurons, a process called neurodegeneration, in a very specific brain region. It's deep in the center of the brain, and it's a set of cells that are normally responsible for sort of repressing excessive motor activity. And so when you have Parkinson's disease, it leads to this syndrome associated with muscle tremors and eventually sort of difficulty walking and carrying out normal motor functions. So it seems like if you have a disease like Parkinson's that's caused by the death of specific neurons in the brain, well, we should be able to just stimulate neural stem cells to divide and replace that cell population that's been lost. But the problem is that these stem cells are dispersed throughout the brain, and we don't have a really good way of targeting just those stem cells that are in that region of the brain that we want to divide and causing them to specifically form the types of cells that are lost in this syndrome that control motor movements specifically. Now, we obviously can't take them out of the brain. If they're widely dispersed in that brain tissue, it's very difficult to find them and actually get them out of the brain and then put them back in where we want them. So it's a bit of a problem. We can't really effectively use the neural stem cells that are already there in that tissue and stimulate them to divide as a potential therapeutic intervention for this disease. So what would work, though, is using something like embryonic stem cells, right? Those embryonic stem cells that we just talked about, which I'm just going to abbreviate now, ES cells, embryonic stem cells, which we know are pluripotent, meaning they can become any type of tissue in the body. If we could get embryonic stem cells from that very early stage of development, about day 5, get those embryonic stem cells out. They're very easily cultured in the lab, so it's easy to grow them. And based on years of research, we also understand, in many instances, how to treat those embryonic stem cells with specific growth factors that will induce them to differentiate along the pathway we desire. So, for example, if we wanted to produce a population of neurons, we would treat with a specific growth factor called nerve growth factor that would stimulate differentiation along that developmental pathway. So for many types of tissues, we know exactly what growth factors to use. We know how to do that. It's pretty easily accomplished. And we can also transplant these cells pretty easily back into the tissues that we want to try to have some therapeutic intervention in, right? So if it's Parkinson's, we would be able to implant these differentiated cells back into the patient pretty easily. So the potential is definitely there with using embryonic stem cells to be able to come up with therapeutic interventions for these diseases to replace damaged tissue or cells that have been lost and potentially relieve the symptoms of the disease. So in theory, this kind of an intervention should be relatively straightforward, right? However, in reality, there are some problems that need to be overcome. One of the problems is political. So there's some resistance to using this sort of an intervention, embryonic stem cells, because of the source of the cells themselves, because we're actually taking those from human embryos. Even though they're human embryos at a very, very early stage of development, there's some controversy, some ethical discussion about whether that is a path of treatment that should be pursued or not. So that's definitely one issue. But aside from that, there are biological issues that have to be overcome as well. So if you think about all the things that have to happen, so if you've gotten these cells out, you've treated them with growth factors, you've gotten the cell types that you want, you've put them back in the body, even beyond that point, all the things that have to go right in order for this to work, those cells have to be able to survive in the body after the transplant. So you're no longer feeding them nutrients and growth factors, and they have to be able to survive on their own. They have to be able to integrate effectively into the tissue. They have to become a part of the tissue. And remember that this is a process that normally occurs, how that tissue is organized early in development. So this is not a trivial feat that we're asking for these cells to accomplish here, to jump in after the fact and replace those cells. They have to be able to continue to function correctly long term. So if this is going to be a long-lasting therapy, then these cells have to persist and they have to retain their properties and their correct functions long term. A big one is that in carrying out this kind of a therapeutic intervention, we have to avoid harming the patient. And the biggest source of potential harm is that we are taking stem cells, manipulating them, and putting them back in the body where they weren't before. Now, ideally, if you take stem cells and you treat them with growth factors, you cause them to differentiate, they've all differentiated. But what if you implanted back into somebody's brain a population of differentiated stem cells that contained just a few stem cells that hadn't differentiated? Well, those cells are capable of dividing, and if they start dividing out of control, then that can lead to the formation of tumors. So that's a big risk that is associated or potentially associated with the use of embryonic stem cells as a therapeutic intervention. And finally, we have another problem with using embryonic stem cells that's essentially the same problem that comes from using transplanted organs, and that's the potential for immune rejection. So these are cells that did not originate in the patient's body, and they'll be recognized as foreign, and the immune system will mount a response and attempt to get rid of that foreign agent from the body, and that's a rejection response that's also associated with organ transplantation. It's no different here. You have foreign cells being rejected by the body. So all of these potential pitfalls have to be addressed in order to become a routine intervention in the clinical setting. A recent avenue of research that's been pursued attempts to overcome both this potential problem, immune rejection, and this potential problem, the political sort of ramifications of embryonic stem cell research and therapeutics. And this strategy relies on the use of what are called induced pluripotent stem cells. Now, this is something that's only been investigated within the last few years. The first publications that demonstrated that this was possible were in 2005, so this is a really recent development in the research community. But what this is very nicely described by the name of this population of cells, induced pluripotent stem cells. So they're pluripotent stem cells, right, just like embryonic stem cells, but they're induced to become pluripotent. So they don't start out as being pluripotent. They undergo some modification that leads them to be pluripotent. And to produce this population of stem cells, what you would actually start with are cells from the patient themselves, and these would be adult differentiated cells. So you could actually take a sample of cells from skin, for example. And then what you would do is essentially turn back the clock, and you would use genetic tricks to express that set of transcription factors that's normally expressed in embryonic stem cells. ES cells. So remember in our discussion of embryonic stem cells and what made them pluripotent, and what the difference between those embryonic stem cells were and the differentiated cells of the germ layers, is that they expressed different sets of genes. The transcription factors that are expressed in embryonic stem cells keep them in an undifferentiated state. So with this method, what you do is you start out with differentiated cells. You utilize techniques that allow you to express transcription factors that are normally expressed in the undifferentiated cell type, and you turn back the clock and you cause these cells to revert back to a state that's similar to embryonic stem cells, back to a state where they're pluripotent, right? You go from a differentiated adult cell type back to an undifferentiated pluripotent cell type. And now you do the same thing that we just talked about for embryonic stem cells. You treat those isolated cells with the appropriate growth factors that will lead to differentiation of those pluripotent cells along whatever developmental path you're interested in pursuing. So you select the growth factors depending on the result that you want to get. Now, the advantage of using this technique is that the cells came from the patient themselves. So, going back to our Parkinson's example, what you would do in theory, right, this again is a very new line of research, this hasn't been done yet, what you would do in theory is take skin cells from that patient, turn back their clock, induce them to become pluripotent, treat them with appropriate growth factors to get neuronal cells, and then you could take those neuronal cells and transplant them into the brain of the Parkinson's patient, into that brain region where the cells have died, and potentially alleviate that cell death and that neurodegeneration that's occurred. Because these are the patient's own cells, you won't have any immune rejection, and because we're not starting out with embryonic cells, there's no political ramifications for this method at all. And so this has been shown to work really well in animal models. It's now been shown to work with some types of human cells. A lot more work needs to be done to make sure that this process occurs the way that it should. But the thing to recognize is that all of those other issues that we just talked about for embryonic stem cells, so getting them to survive after they're transplanted, getting them to integrate, making sure that they function correctly long term, and avoid harming the patient, all of those are still going to be at play. So we've addressed a couple of problems, potential problems with this technique, but certainly not all of them. So even though this method holds promise, we're still a long way from being able to use that in the clinical setting. Because embryonic stem cells have been the subject of research for a much longer period of time, they're actually much closer to being used as a potential therapeutic than induced pluripotent stem cells. In fact, there are clinical trials already underway for embryonic stem cells for treating various diseases, things like diabetes, to replace the cells that produce insulin that are lost in diabetic patients, as well as a disease called macular degeneration. This is a common cause of blindness in the elderly, and it's caused by the death of neuronal cells in the back of the eye. These are called retinal cells, and so the stem cell therapy would be aimed at replacing those retinal cells and preventing further loss of vision from occurring. So we're actually closer to therapeutic interventions with embryonic stem cells than we are with induced pluripotent stem cells. So we've gotten to the point where we can, with some level of efficiency, replace worn out cells or dead cells with the same cell type, right, derived from embryonic stem cells. Ultimately, an important direction that this field is going is in what's called regenerative medicine, in which whole organs, right, we're talking complex organs, things like kidneys, things like a whole heart, might be able to be engineered in the lab to replace worn out organs, right? So eventually we could get to a state where maintaining your health is sort of like maintaining your car. If your heart wears out, you go into the mechanic and you replace it with a new one.
Now that we've completed our discussion of stem cells, you should have a good understanding of their normal roles during embryonic development, as well as in maintaining the health and physiological function in the adult, as well as a better understanding of the potential therapeutic applications of stem cell therapy. Now that you've watched this video, you should be able to answer these concept check